We might be just weeks away from knowing whether a new antibody drug can be effective in the fight against COVID-19. I'm Lee Kelso. This is HealthCall Live Online, where we offer extended versions of interviews featured in our weekly radio broadcast. And today we're talking with the Vice President of Eli Lilly, Dr. Ajay Nirula. He is the medical team leader for development of a new antibody-based drug for COVID-19. You're a busy guy. Appreciate you taking the time. Thanks for joining us.
My pleasure.
So help me understand here. We've got antibody therapy for Ebola and other viruses. How does this particular antibody drug attack this particular virus?
Yeah, so it's been an interesting process the last couple of months as we've developed this antibody against the SARS CoV-2 virus to help patients with the COVID-19 illness. The antibody responses are an important part of how the body responds to viral infections. And so when this pandemic sort of overtook the planet earlier this year, Eli Lilly was very excited to get involved in helping with potential therapies. And what we did was we took blood from one of the first recovered patients with the viral infection, and we basically isolated the antibodies that the patient was normally making against the virus, and we're able to sequence and profile these antibodies in detail. This was done as part of a collaboration with a small biotech company called Abcelera, based in Canada. And a lot of our scientists at Eli Lilly spent long days and nights working on these antibodies, characterizing them, engineering them, to the point we had some very good antibodies against the virus that we could measure by a variety of assays. And basically, we then took the best antibody and really started to produce it to be ready for clinical trials. And we achieved that last week by entering the first antibody into clinical trials.
That our own body creates, we're the source of this. How are these antibodies different when they're created externally as a medication? Do they last as long within the body?
Yeah, so when we make an antibody response, to an infection or a virus, our body makes lots of different antibodies. So we literally could have hundreds of antibodies trying to fight the infection. It's what we call a polyclonal response. But what we did was to sort of hone in on what we think are the best antibodies for fighting the virus and make a lot of them. So when we give this antibody as a medication, it's really an enriched sample of the best antibody. And we've started with one antibody, but we have several good antibodies against the virus that we may end up bringing into the clinic soon in combination also. So.
What is it doing? How is it blocking the virus?
Yeah, so different antibodies can bind to different portions of the virus. We've focused in initially on antibodies against what's called the spike protein of the virus, and the spike protein is a critical part of the virus that helps it enter cells. So the antibodies that we've engineered and put in the clinic can bind the spike protein. It can stop the virus from binding to the receptor on the cell surface and entering the cells, but also the antibody has other functions that go with it. So it can actually also mediate killing of the virus or of infected cells. So that's some of the things that antibodies normally do when we fight infection. But with what we've put together as sort of one of the better antibodies for binding the virus, that's what we hope it will do as a medication.
Described as this spike protein unlocks the key to the cell. And do I understand that this antibody sort of gums up that process and keeps that from happening?
Yeah, it's sort of blocking the key going into the keyhole, if you will, and blocking the whole process of viral entry. But then it alerts the rest of the immune system that there is an invader, and then the rest of the immune system can come in and clear the virus.
Does the body then begin producing antibodies similar to this, or is it just, as you said, that kind of alert, hey, there's something going on, you need to respond?
Well, so yeah, when a body normally, when a person normally fights an infection, it's making lots of antibodies. That's one way it fights an infection. It also has specialized cells of the immune system that can fight an infection and some other immune type functions. So they all work together, and antibodies, work together with the other pieces of the immune system. Now, of course, what we put into the clinic as a medication is actually an antibody that was originally produced by one of the survivors of the infection, and so it's very similar to one of the normal antibodies we make. We make some subtle changes to make it make it a better drug in terms of how long it stays around in your system and so on. But in the end of the day, it's similar to an endogenous antibody. It's just that with the medication, we're really giving the best antibody or best mixture of antibodies down the road that will help fight the virus.
Injection therapy, but tell me about how it's being used in your clinical trials. So you're in 3 different medical centers, 3 different hospitals. Yeah. I know it's the typical placebo, double-blind. You don't know if you are actually receiving the drug, so that you can't really fudge the results in any way. But what kind of patients are receiving it? Are these the sickest of the sick, or where is it coming along in the disease process?
Yeah, so this is what we started last week was the first study, which is what we call a phase 1 study. So it's a relatively small study. In this particular case, we chose to go into hospitalized patients first, and we did start off in three well-known medical centers. We're expanding to others. And it's a relatively small study on the order of about 30 patients. And we will, the primary goal of this first study is to get safety and tolerability data for the drug. We'll also understand the pharmacokinetics. So that's, how the drug behaves in the body and how long it stays in your blood and so on. We'll get all that from this first study. We will have what we call some exploratory endpoints looking at impacts on levels of the virus. But ultimately, what we need to do is get to bigger studies, and we will be doing that quite soon, actually. And those bigger studies will be in a variety of populations. So we started in hospitalized patients, but we will be going into the outpatient setting, and we're going to focus on patients, relatively soon after they get the infection, because we think that's when the antibody could have the best impact, is if you can treat early. So we'll be starting those other studies relatively soon, and they'll be all over the country. And ultimately, we'll go to other countries also.
Well, that makes sense. So if the focus of this antibody therapy is to avoid the spike protein from allowing the virus to enter the cells, so what you're really doing by giving it early in the disease process is reducing the overall viral load, right? You're cutting back on the amount of virus my body has to fight. That makes a lot of sense why you'd want to give that early. What are you looking for in these initial patients? Are you expecting them to have some recovery effect? What are you looking to see?
Yeah, I mean, it's a great question, and it's a little bit of an unknown. Again, because this is a relatively small study, it'll be hard to make any definitive conclusions. And as I said, the primary goal is to make sure that the drug is safe, well tolerated, that it behaves as we expect it to, and we'll hopefully over time get to see the overall effect of the drug when we have larger numbers of patients, and it's a better scientific experiment that day. I think with these initial patients, again, we're just hoping to see that the drug is well tolerated and it's safe and it gets to the levels we hope. We will look with great interest at the levels of virus, which we can measure by an assay called PCR. So we can look at the viral load, and we'll look sort of in an exploratory fashion at what impact we have on those. It's a little hard to say right now, but in theory, you would hope that this antibody can help bring down levels of virus, and we'll take a look at that, but with very small numbers in the first study.
So the next study, as you said, more hospitals, more patients,
Yep.
And eventually then, if this drug is released and distributed for use, where do you said you want to give it early, but where in the disease process do you think that would be? Would it be in an outpatient setting, or would I have to be hospitalized to receive it?
Yeah, so I think the hope is that you could intervene early. And so that would, ideally even be in the outpatient setting before patients get very ill. So soon after somebody had a positive test for the virus, they'd be able to come in and get a medication like an antibody. And Basically, hopefully you could treat them early enough that they don't get very ill and progress to the hospital. Having said that, I mean, there could be a range of uses for this, and we'll need to do the clinical studies to understand that. We're not ruling out that this could help later also, because ultimately some patients do get very ill and they go into the hospital, and there are going to be a number of different types of medications that are evaluated to treat these patients. So there's already antiviral agents like remdesivir that are in use. Antibodies could be an important part of it. And then some of those patients who go into the hospital, are getting immunomodulatory medications, because a lot of the reason that they're getting ill is that there is a profound inflammatory response to the virus. So you could envision all of these medications could theoretically work together, but it remains to be proven. Another setting where this could be used, and you brought up the analogy earlier of this blocking the key, turning the lock, if you will, is you could give this prophylactically, and it could theoretically block infection. So, as we wait for good vaccines to be developed, there is some potential use for antibody therapies in prophylaxis. I think we're all hoping for the companies who are working on vaccines to succeed and for the world to have good vaccines as soon as feasible. But in the meanwhile, I think there could be a use for this antibody therapy for prophylaxis. And even after vaccines are developed, vaccines are not always completely effective. So there could be a use for these types of antibody therapies in special populations who don't respond well to vaccines or who have special medical conditions.
Or in the prophylaxis, I assume you're thinking of high-risk groups, nursing homes, immune-compromised, those kinds of folks, right?
Absolutely. Those are the patient populations we're thinking of. And we've watched with sadness what's happened with the pandemic, and how institutions such as nursing homes have been profoundly impacted. And so this, in theory, would be a good medication to give prophylactic in that type of setting. And the elderly and folks with comorbidities that increase the severity of the illness will also be, considered for prophylactic type studies.
About how you develop a drug like this. So you said earlier that you start with convalescent plasma, taken from someone who's recovered from the disease. And then what are your guys doing in the lab, and what's that process like?
Yeah. It's very close to what you described. When we get blood from one of the recovered patients, what's being done with in some studies is to actually take the plasma that has the antibodies and use that therapeutically in other studies. What we actually did was we took the cells that make the antibodies. So these are special cells called B cells or related cells, and we can actually get the actual DNA sequence of the antibodies. So it allows us to make the antibody or make something really identical to the antibody that the recovered patient made. So once we get that, we can express these antibodies in cell lines in the labs, make a lot of the protein, and then start to characterize the protein. So I mentioned earlier the spike protein. We studied hundreds of antibodies, how well they would bind to spike protein, for example, and then we picked the very best and went even further. And we did a lot of different assays, showed its binding properties to the spike protein, but actually characterized its ability to prevent the virus from infecting cells and replicating. And that even involved experiments with live viruses, which we did collaboratively with a lot of the top labs in the world. And we would take our antibody and test its ability to block the replication and spread of the virus in some of these live virus assays. Ultimately, we will also get to experiments in animals. In fact, some of those are underway, but there are animal models of viral infection that we'll be assessing our antibodies in. Normally, in drug development, you do a lot of this work over the course of years before you took a drug into the clinic, but this is a very special circumstance with the COVID pandemic. So we wanted to get the drug to the clinic as fast as possible, just because patients need it, and we're doing a lot of this work in parallel.
Yeah, it's real interesting how the system, the entire environment in which you work has to have just gone through a revolution in the speed in which we're approaching all of this. How is that going to transfer or be applied to other projects you're working on? Are you going to have that kind of flexibility, you think, going forward in whatever other condition you're trying to treat?
I think this is this experience with the COVID pandemic and developing therapies shows how quickly we can do things when when there is an urgent need. And even though I don't suspect we'll do it this way for every drug we develop in the future, I think we will definitely take efficiencies from this and apply it in the future, because it will be a nice outcome of this if we can actually get drugs to the clinic quicker. I mean, a lot of the reason we do take a lot of time to develop drugs carefully is first of all, we're very concerned about the safety of our medications, so we want to make sure those are tested optimally before they go into the clinic. And sometimes it's not always clear which are the right ones to take to the clinic, so we have to do a lot of experimentation. But this was a unique situation with COVID that we knew we wanted something to target the virus. We had a pretty good sense which protein we wanted to target, and the need in the clinic was so acute that we found the best balance between speed and maintaining safety. So that's I do think your question is has a correct premise to it, that this will change a lot of drug development in the future, and things will hopefully get more efficient in some ways.
Well, that is exciting. So I just am curious about how you create this stuff in large quantities. So you've taken the DNA of the protein required to create this antibody, and is it like a giant kitchen? There's big vats. I mean, not to simplify it too far, but help me out with that.
It is. I mean, if anyone's been to a brewery and seen those giant vats where beer gets made, we have these giant incubators where we grow cell lines. They're not always human. In fact, one of the common ones we use is a hamster cell line called CHO cells. But we can express the protein of interest in these cells, grow them in large cultures, and then purify the cells, purify the proteins. It's a pretty impressive process to generate a lot of drug in a short time. And with the acute need in the clinic, Lilly has some great manufacturing capacity, for antibodies, but we've really turned a lot of our focus into making medications for these clinical trials.
And how much of this stuff are you working on? My understanding is that you're pretty confident and that you're already into production.
Well, we're making a lot of material for the clinical trials, and we're thinking ahead in case these medications succeed. If they do succeed, we want to be ready to meet the needs. So we are making a lot of medication at risk that can be used.
So if it works, if your final stage of the clinical trial would be about when and how soon could you submit it for approval and actually get it into the clinic?
Yeah. Basically, we're starting off with the phase 1 studies, which take a matter of weeks, and pretty quickly getting into the phase 2 studies, which take, we think will take a few months. Ultimately, we'll hopefully get a signal as to whether the drug is working in those phase 2 studies, and then we'll look at, based on the data, we would go to more phase 3 studies, which again, we're trying to move as quickly as we can. So hopefully that'll be over the course of a few months. So I think we're sort of looking into a few months down the road to have some meaningful results, and then we'll see if the data justifies seeking approval for the drugs for COVID therapies. We're obviously hopeful that some of these antibody therapies will work.
I appreciate your need to be a little vague on that. I get it. I understand you have to walk that line very carefully. So let me wrap up with this. Is there anything I didn't ask you that you want the public to know about what Lilly is doing?
Yeah, I mean, I think what I'd like to say is that Lilly is really committed to trying to make an impact on this global pandemic, and it's a wonderful example of teamwork across the scientific world. So whether it's universities, medical schools, or the government, institutions like the NIH, or our peers in the pharmaceutical and biotech industry, everyone's working, together to try and make an impact. And I've never seen such a tremendous example of collaboration across the industry. And I think we're all rooting for joint success. So I think there is a lot of collaboration, a lot of folks working together. Ultimately, I think with therapeutics or with vaccines, I'm hopeful that as the better medications emerge, the industry will get behind what look to be the best medications and help the companies who develop those push those forward to the finish line. So it's really been a tremendous example of collaboration across the scientific world, which includes our sector.
Okay, so I misled there just a moment ago. This is the final question, and it's kind of a silly one, but I think a lot of people will be interested. So the nickname for this drug, I understand, is Lucky 555, and you have an internal name for it. But when it goes to market, how do the names of these drugs come about? I mean, we've got, leronlimab and all those crazy names that people hear all the time, Intruda and other things. Who comes up with that stuff?
Yeah, I mean, so some of the names, the generic name, have a relatively standard format. So antibody therapies, for example, there is an international committee that guides the generic naming of medications, and so monoclonal antibodies often end in MAB, for example. But then the trade names are sort of come up. The companies have groups that are very good at coming up with the names that I think capture what the vision of the medication is, and hopefully make it easy to remember for patients and prescribers. So there are specialized groups in our commercial functions who do come up with those names.
Well, listen, I think many people join me in wishing Eli Lilly the best of luck with Lucky 555. We hope we see it in production, in use, and helping COVID patients very soon. I thank you for joining us. Ajay Nirula is the medical director, or the team director of the medical team leading development of this drug, and he's also vice president at Eli Lilly. Thanks again.
Thanks for having me.